So hi, everyone. I'm Kristin Burnum-Johnson, and I am a senior scientist at the Environmental Molecular Sciences Laboratory, or EMSL. And in this presentation this morning, I'm going to discuss proteomics. First, I'm going to give an introduction by highlighting some basic concepts. Second, I will introduce important concepts that Yu Chin and Yvette will discuss in detail in their presentations later today. And third, I will highlight some of the cutting edge ongoing proteomic research in EMSL. So by the end of today, all of you watching are going to be proteomic experts. So yesterday, we introduced the concept of design, build, test, and learn, or DBTL cycles for synthetic biology research, where we're optimizing the genetic engineering of microbes to make high titers of platform chemicals that can be used for industrial biotechnology. And for example, we are engineering Aspergillus pseudoterius to make large quantities of 3HP. 3HP is a valuable platform chemical, which means it is a precursor in the industrial production of a number of chemicals such as acrylic acid. So bioplastic production is one example. Therefore, instead of making plastics with petroleum-based raw materials, we can use renewable materials, reduce the use of fossil fuel resources, have a smaller carbon footprint, and faster decomposition. Now, for this research, proteomics is an important part of test. And throughout today, you will be hearing about the essential components of the proteomic workflow pictured here in the colored boxes. So sample preparation in orange, separation in red, mass spectrometry in pink, and data analysis in purple. So let's start with the orange box, sample preparation. So sample preparation methodologies that will be discussed in more detail today include trypsin digestion of proteins into peptides, specialized enrichment techniques for post-translational modification detection, such as phosphoproteomics, both nano and micro well sample processing for small samples that can even look at single cells, and utilizing isotopically heavy C13 peptides for targeted proteomic measurements. Now let's move to the red separation box. So today you will learn about online and offline separation methods to decrease sample complexity prior to mass spectrometry detection. And there's a few keywords highlighted here. So high performance liquid chromatography, HPLC, using microflow and nanoflow, and also electrospray ionization, which is used to introduce ions into the mass spectrometer. For the pink mass spectrometry box, mass spectrometry methodologies that will be discussed in more detail today include bottom-up and top-down proteomics, targeted proteomics, ion mobility, and Fourier transform ion cyclotron resonance, or FTICR, data-dependent acquisition, DDA, and data-independent acquisition. And for this last box, for the purple data analysis box, mass spectrometry data analysis techniques that will be discussed today include MSGF+, MaxQuant for discovery proteomic pipelines, Skyline and selected reaction monitoring, or SRM, and data independent acquisition and ion mobility analyses, and other software tools such as OpenSWAT or FragPipe. So by the end of today, you will have a detailed understanding on how all these components, or as pictured here, pieces of the puzzle essentially fit together for a comprehensive proteomic workflow. Now, yesterday, I already showed some slides detailing all of our mass spectrometry instrumentation, but I want to highlight our mass spectrometers used for proteomics again today. For discovery proteomics, we predominantly use thermal Orbitrap instruments. For targeted proteomics, we predominantly use triple quadruple instruments. And for advanced proteomic measurements, we leverage the enhanced separation of ion mobility and high mass accuracy of Fourier transform ion cyclotron resonant, or FTICR, measurements. So proteomics is a high throughput approach to quantify protein levels, but these measurements enable not only the presence and abundance of proteins, but they can also provide an understanding of the structure, the function, and the interactions of proteins. So if you look at this figure, the proteome circle in the center of the slide depicts how you can study the proteome in biological systems. You can study the abundance of individual proteins represented by the pink, blue, and purple protein structures to the left of the circle. You can study small molecule post-translational modifications or PTMs such as phosphorylation or acetylation. And you can study large molecule PTMs such as glycosylation and ubiquitination. And you can study protein complexes. Now, everyone, for your questions, I see a few popping up. Keep putting them on the comments there. And when this is complete, we'll go ahead and go through all of the questions that you guys have. So keep them coming. So this slide, it captures the flow of information. So we're all familiar with this: DNA or genes to messenger RNA to proteins to metabolites, such as those depicted here on this slide. So carbohydrates, lipids, and amino acids. So we go from the genome to the transcriptome to the proteome to the metabolome. And when we study the proteome, we're interested in protein content, protein activity, and post-translational modifications. So the DNA or genes are what could be, they're the blueprint. The RNA is what it's trying to be, and the protein is what it is. Therefore, if we want to understand function, it's important to study the proteome and the metabolome. So what do I mean when I say the protein is what is? Well, proteins are physiological active key players. So here we see our DNA, messenger RNA, and proteins. The proteome is the entire complement of the proteins expressed in a cell, a tissue, or organism at a given time. So the proteome is dynamic. It changes in response to stress, in response to treatment, in response to environment. And transcriptomics is a very powerful tool, but often the transcriptome correlates only loosely with protein expression due to post-transcriptional and post-translational control mechanisms. So let's discuss proteomics as a tool for synthetic biology. Putting this in context with our Aspergillus pseudoterius. So again, our goal is to make high amounts or titers of 3-HP. We design, we build, and then we use our proteomic workflow to test a suite of engineered mutants. This results in us knowing the protein abundances for each engineered mutant. And we also quantify the response or titer of 3-HP for each engineered mutant. Now, we typically determine the relative protein abundances, but in cases where we need higher accuracy, we can use isotopically heavy peptide standards, which can give us close to absolute abundance. Now, with all this data in hand, we use Learn to predict what additional engineering mutations we should design and build to further increase 3-HP production. And as you will learn later this week, putting this protein information into kinetic models of metabolic pathways with complementary metabolite data. So if you have proteins and metabolites, is really a powerful technique, which enables us to make precise predictions. We can predict where we need to delete, where we need to increase protein expression to increase 3-HP production in the next design and build cycles. Then we test and we learn from these next generation mutants until we get a fine-tuned Aspergillus pseudoterius strain, which is using all this energy to make our target molecule 3-HP. Now, going to basics, everyone's familiar with this, but so we're particularly interested in proteins that serve as enzymes, which are catalysts for these biochemical reactions of interest. So proteins are the workhorses of cells. On a molecular level, they're made up of linear chains of amino acids, as pictured here. So each amino acid contains a central carbon atom, an amino group shown in blue on this slide, a carboxyl group shown in red, and a specific R group shown in green. So the R group determines the characteristics, the size, the polarity, the pH for each type of amino acid, which I'll mention on the next slide. So peptide bonds are formed by biochemical reactions that extract a water molecule as it joins the amino group of one amino acid to the carboxylic group of the neighboring amino acid. So we all know amino acids are the building blocks of proteins. Now, these amino acids, they fall into different categories where they're nonpolar, they're polar, they're basic, they're acidic. Protein structure really depends on the amino acid sequence. So the protein structure plays a key role in its function. And if the protein loses its shape at any structural level, the primary, the secondary, the tertiary, it can lose its function. So for example, if you have a transmembrane protein that's involved in transport, that inner part that's part of the layer within the membrane, it has a bunch of nonpolar or hydrophobic amino acids because it needs to match the chemistry of those carbon chains that are made up of the fatty acids that make up that bipolar layer. So, the structure depends a lot on these amino acids and what they look like in terms of chemistry. Now, there are multiple approaches for the assessment of proteins. Circled on this graph, there are antibody approaches to the left, as you see, and fluorescent protein approaches in the bottom right. Now, I'm not going to spend a lot of time going over these other approaches in detail because our synthetic biology studies, we want to quantify a lot of proteins with high specificity, and mass spectrometry-based proteomics can do that, especially bottom-up proteomics, which is in the top of that blue mass spectrometry box that you see in the upper right. So with bottom-up discovery proteomic approaches, you can identify and quantify thousands of proteins in a single experiment without any prior knowledge on the proteins you're interested in, which makes this a very powerful discovery tool. So you will be learning more about bottom-up proteomics today, since as stated in that sentence down at the bottom of the slide, bottom-up proteomics, it has a high number of quantified proteins and high protein detection specificity. So although bottom-up proteomics will be our main focus, we will also discuss top-down proteomics for intact protein measurements and in situ measurements, where sampling is done directly from the surface of the sample, such as with MALDI or matrix-assisted laser absorption ionization measurements. So throughout the day, we'll be covering multiple proteomic methods at a high level to introduce the concept, or at a real in-depth level for methods that we routinely use that we want to emphasize. So with the high number of quantified proteins and the high protein detection specificity with bottom-up proteomics, we are able to visualize our molecular pathways and comprehensively understand the molecular flow of information. So this slide captures many pathways of interest, including, you see, glycan biosynthesis and metabolism, nucleotide, carbohydrate, amino acid, and energy metabolism, biosynthesis of polyketides and terpene molecules, and xenobiotic biodegradation metabolism. So a lot of different things going on. Pathways of interest, they change according to our particular microbial hosts and in our particular target molecules we are interested and that we're trying to produce in large quantities for industrial biotechnology applications. And also different design, build, test, and learn cycles, they affect different pathways because introducing mutations to try to increase the titer of our target molecules, it can often have unexpected consequences. For example, if a biological pathways, they often have redundancies. So deleting an enzyme might not have the desired effect because the host could compensate by initiating expression of a different enzyme that has complementary functions. So we see this quite often. It's a battle between us and the microbes to try to get them to increase titers of those target molecules. So multiple design, build, test, and learn cycles and bottom-up proteomic approaches, They provide detailed views into how these pathways respond. So really, for our analyses is bottom-up proteomics for the win by having the ability to capture this information. So it's important for us to build our predictions and pathways based on proteomics as you move from the genome to the transcriptome to the proteome. As many of us are aware, there's an increase in complexity. So if you look at examples highlighted on this side, if you start with 21,000 protein encoding genes, then there is alternative splicing, alternative promoter usage, messenger RNA editing. So now you have 100,000 transcripts. Then you add on post-translational modifications, and now you could be looking at 1 million proteins. So to emphasize this point, the proteome is complex. We're getting an increase in information as we move from genes to messenger RNA to protein to proteoforms. And measurements need to be made at the proteome level to really capture this complexity so we do not miss important molecular information. And that is why, when needed to complement in-depth traditional discovery proteomic methods, which look at changes in relative abundance, additional measurements can be performed with emerging technologies to characterize post-translational modifications at the peptide or protein or proteoform level. Most often our traditional bottom-up proteomic methods, they provide information we need for our design, build, test, learn cycles. But when we need to characterize site-specific features to address specific biological questions, we do have advanced instrumentation capabilities here at EMSL. So our emerging technologies can help with these specific questions. Now, in addition, or I guess an additional reason it's beneficial to make measurements at the proteome level is environment and feedback mechanisms also create knowledge gaps for function, where protein and transcripts levels often do not match. For feedback mechanisms, you have metabolites, proteins, and messenger RNA counts or their abundances change the rate of transcription or translation. Changes in environment, for example, for a lot of our systems, changes in pH in our synthetic biology cultures can initiate stress response and consequently alter the rate of transcription, translation, protein turnover. Protein levels have significant effects on biological function and phenotypes. Therefore, the cells actively use different regulatory mechanisms to control protein abundance. A common and well-characterized negative feedback mechanism is protein-mediated transcriptional regulation, where the protein expression from a gene actually inhibits its own transcription. For example, it's estimated that over 40% of E. coli transcription factors can regulate their own expression through this feedback mechanism. So really, there's an inherent complexity with all this molecular information. And with proteomics, we have the ability to capture this complexity that exists in these systems. So, just to simply state it, we can go beyond a parts list, as you're seeing there in the left, to understanding interactions, regulatory, enzymatic, and signaling interactions, and then, with our goal to obtain a detailed view of biological networks so we can truly understand our systems and help in that design process. Now, I want to start discussing some more details about our proteomics workflow. So this slide depicts our MPLEX protocol for single sample proteomic, metabolomic, and lipidomic analyses. If you look at the picture on the right, the top metabolite and bottom lipid fractions are removed for metabolomic and lipidomic analyses. Then the proteins, or that protein disk that you see there, is further processed. Now, in previous slides, I kept mentioning bottom-up proteomics. Now I want to discuss this approach in a little more detail. In bottom-up proteomics, mass spectrometry measurements are made at the peptide level, which means we need to digest this protein disk. As you see in the bottom digest box on this workflow, we resolubilize the protein pellet with urea, which denatures the protein. So when the protein is denatured, its loses its secondary structure. So it loses its tertiary, secondary structure. So it's just a protein that becomes unfolded, and now it exists as a linear string of amino acids. Then trypsin is added to make peptides. Since the protein is denatured, trypsin can easily cut proteins into peptides at lysine and arginine amino acid residues. Now, in traditional bottom-up proteomic sample preparation, for our Aspergillus pseudoterius samples, we homogenize frozen fungus pieces prior to MPLEX to ensure efficient cell lysis. Those of you that have worked with fungus before, it has that rigid structure outside, so the homogenization helps with that. So we take our tissue, we homogenize, and we extract the proteins. Then we denature, reduce, and alkylate. Reduction and alkylation of the disulfide bridges, it keeps the protein denatured. Then we digest our proteins with enzymes, for example, trypsin that I keep mentioning, but there are other enzymes that you can use too. Now to make peptides, so once we have our peptides, then we desalt our sample so it's compatible with mass spectrometry analyses. So we take our protein solution here, digest it into a peptide mixture. Here we have trypsin, and following trypsin digestion, the majority of the peptides are an ideal size for mass spectrometry. Therefore, these peptides are then well characterized by liquid chromatography, electrospray ionization, mass spectrometry, and this will really be covered in more detail later today. Now, although I have so far emphasized bottom-up discovery approaches, I did mention that when needed to address specific biological questions, we have advanced measurement capabilities here at EMSL. So we do have additional options that you will learn about today. And as stated at the bottom of the slide, it is always the biological question that drives the approach. So following protein extraction, instead of digestion into peptides, you can leave your protein intact, or you may isotopically label your peptides for multiplex analyses or enrich a specific subset of peptides. If you go to the next one, for separation prior to mass spectrometry analyses, you may fractionate. You may choose to use a long or short gradient for in-depth or fast analyses. And then in the mass spectrometer, you can fragment your ions in different ways. You may have label-free or label peptides, or you may generate proteomic data using data-independent acquisition, DIA, or data-dependent acquisition, DDA. So we have lots of choices here. And as I mentioned, it's really the biological question that drives the approach. And sometimes novel approaches are needed. So I want to highlight some of the cutting edge ongoing proteomic research at EMSL. On each one of these slides, I've included the contact information for an EMSL scientist that's really driving these cutting edge technologies and applications, so they can be contacted later. So on the next slides, I'm going to introduce, as depicted here, high throughput gas phase separation of ion mobility separations, miniaturizing sample processing to nanoliter volumes to enable proteomics on small samples, and also analyzing large intact molecules such as top-down proteomics for intact protein measurements to understand proteoforms and how the different post-translational modifications affect one protein. So this slide depicts our top-down intact protein measurements. If you look at the right of the slide, bottom-up proteomics, it states it's well established. We know it's a high throughput pipeline that analyzes peptides. It works well for the majority of our agile biofoundry applications, but as an emerging technology, top-down mass spectrometry visualizes the combination of PTMs on a single protein. And also, native mass spectrometry captures non-covalent interactions, such as complexes, to visualize ligand binding. Now, to visualize proteins and native protein complexes in a high throughput manner, We have a matrix-assisted laser desorption or ionization or MALDI platform that's coupled to an ultra-high mass range Orbitrap. So currently, we can detect proteins up to 40 kilodaltons, and we are optimizing for even higher masses. So MALDI mass spectrometry analyses are performed directly on the sample. MALDI is an ionization technique. It uses laser energy absorbing matrix to create ions from large molecules with minimal fragmentation. So the sample is placed on a charged plate, either metal or coated glass, is coated with matrix. A pulse laser irradiates the sample, triggering ablation and then desorption of the molecules, which are then detected and quantified by mass spectrometry. Now, analyzed samples can be aliquots of cell extracts, or as depicted in this example for this slide, it can be actual microbes or multiple microbes. And what's really nice is by tracking the X and Y coordinates of each laser ablation, you can create two-dimensional images. So that's multi-imaging mass spectrometry. And we can use this imaging approach to map microbial interactions across both space and time. Now, for sample-limited proteomics, EMSL developed a novel nanoscale sample preparation, which is being done with nanopots. So nanopot stands for nanodroplet processing, and one pot for trace samples. So with nanopots, all the sample processing steps required for a bottom-up proteomic workflow are performed in nanowells with 200 nanoliter reaction volumes, which decrease sample losses due to nonspecific binding to surfaces and increased digestion kinetics. So in the picture to the left here, you can see the nanowells with 200 nanoliter droplets of colored liquids, so you can visualize them. And in the video to the right, you can see the automated dispensing system, which fills and extracts liquid in the nanowells. Therefore, we now have a robust way to perform all the proteomic sample preparation steps, such as you see here, pictured at the bottom left, our extraction, reduction, alkylation, enzymatic digestion. Here we're using lyse and trypsin, and cleanup all in 200 nanoliter volumes. And if we look at this table here, we can see that compared to a traditional tube method, our nanowell method, we have decreased our reaction volume from 100 microliters to 200 nanoliters. We have reduced our surfaces by 99.6%, thereby decreasing sample losses due to nonspecific binding. And we have increased our digestion kinetics by 500 times. We have also developed a high throughput, large scale targeted proteomic quantification methods, where we apply these methods to our synthetic biology agile biofoundry samples. And there's the reference publication there. So in this particular example, we quantified 132 enzymes while increasing our analysis speed by 4 times while maintaining high sensitivity using microflow instead of nanoflow liquid chromatography selective reaction monitoring. So Yu-Chin will go over this targeted proteomic workflow and how it compares to discovery proteomics. Details for that she'll cover in her presentations later on today. So we have enhanced proteomic analysis speeds with two-dimensional separations by coupling liquid chromatography, which occurs on the minute timescale, with ion mobility spectrometry, which occurs on the millisecond timescale, and mass spectrometry, which occurs on the microsecond timescale. So Yvette will discuss this more in detail later today too. And another benefit of liquid chromatography, ion mobility spectrometry, mass spectrometry measurements is that applications of our platform separate peptides of interest from high concentrations of environmental organic materials or just smaller molecules that can interfere with our peptide identification. So these usually hurt normal liquid chromatography mass spectrometry measurements. But if you look at the figures, you can see that the LC and the IMS separations of the organic material contaminants, they're actually separated from the peptides. And this aids in the analysis of these particular examples. So leaf material to the left and soil samples to the right. So eye mobility separations is able to move most of these unwanted organic materials or other contaminants to a different drift time space due to their different molecular structure. So as kind of the last part of my talk today, I wanted to detail more of the benefits of a bottom-up proteomic workflow. As just to recap, with the bottom-up proteomic workflow, we extract the proteins, we digest the proteins, we analyze the peptides by liquid chromatography mass spectrometry. We then fragment and identify the amino acid sequences of the peptides. So what are the benefits of the approach? So for our bottom-up proteomic workflow for mass spectrometry, we're leveraging, again, our depicted workflow as I discussed in the beginning of the talk. So sample preparation, separation, mass spectrometry data analysis shown in these 4 color panels to give us deep proteome coverage. So in this bottom-up proteomic workflow, a crude protein extract is enzymatically digested into peptides. Peptides are then separated based on chemical or physical properties prior to mass spectrometry detection and identification. And as you know, our current workflow, we're using liquid chromatography separations. Now on the right, there's 6 advantages of our bottom-up proteomic workflow that enables both unbiased and in-depth detection of the proteome. So let's just take a closer look at these advantages of our bottom-up proteomic workflow on the following slides. So if we look at our sample preparation in detail on the right, we take our samples, we go through our lysis steps for efficient protein extraction and digestion into a peptide mixture. Therefore, our first 2 benefits are number 1, we have highly efficient protein extraction, and number 2, peptides have high ionization efficiency. So we see them well by mass spectrometry. Now let's discuss our separation steps. We're currently using liquid chromatography separations to reduce spectral complexity. As you see in the chromatogram there on the right, these peptide peaks elute over time. Therefore, liquid chromatography separations reduce spectral complexity and minimize ionization suppression. In this workflow, a really important fact is peptides are confidently identified by MSMS or tandem mass spectrometry. In the mass spectrometer, we'll cover this in more detail today, but data-dependent acquisition utilizes cycles of full MS followed by MSMS or fragmentation of the most abundant peaks. Not only do you get confident relative quantitation and peptide identification, but you're able to control your FDR or your false discovery rate, which you want to maintain to be less than 1%. So what are you going to learn today? Well, Yu-Chin will give an in-depth view into how our bottom-up proteomic workflow is used for discovery proteomics, where thousands of proteins are quantified and characterized, and targeted proteomics, where hundreds of proteins are targeted with isotopically heavy internal standards. And this is for really fast and accurate measurements. She'll also go into detail on liquid chromatography and data-dependent acquisition. Now, to expand on Yu-Chin's discussion of liquid chromatography separations and data-dependent acquisitions, Yvette is going to present enhanced separations, where we're combining liquid chromatography with gas phase separations. With this type of approach, we can move to short and fast liquid chromatography gradients coupled to ion mobility, where gas phase separations have the ability to separate and quantitate even co-eluting molecules from fast LCs. So high quality data from really fast analyses. Also, how the molecules behave or where they fall on the trend lines helps identify the molecule type. So molecular class distinction. And if you look at the picture there on the left, you can see the trend lines for the peptides at different charge states labeled in red, and trend lines for different lipids at different charge states labeled in blue. So, you know, we can look at specific types of molecules. Here we're just showing proteins and lipids nicely separate from each other. And then to complement Yuchin's discussion on data-dependent acquisition, Yvette will discuss data-independent acquisition. Now, in the example to the right, the first panel to the left shows data from LC-SRM, so liquid chromatography selective reaction monitoring. The second panel on the left shows data from LC-IMS-DIA, so liquid chromatography, ion mobility spectrometry, data-independent acquisition, which, as you can see, this provides more fragments and thus enables better identification of this particular molecule. So today you will learn more details about sample processing, how to separate and analyze peptides by liquid chromatography, tandem mass spectrometry, and for data analysis, you will learn to search your experimental peptide spectra against theoretical peptide databases using tools like MSGF+, MaxQuant, and Skyline, which are very often used in proteomic experiments. These tools provide peptide identifications, protein identifications, and determine relative and close to absolute abundance when isotopically heavy peptides are used. And for the postdoctoral research and advanced PhD students that will be attending our smaller tutorial classes today, we will spend time analyzing proteomic data from engineered Aspergillus pseudoterius strains with varying levels of 3-HP. And with that, this is my last slide. So I'm going to go ahead and stop sharing and we can start addressing some of the questions. Okay. So it looks like a lot of the questions. Let's go for Melanie's question here. So when you're designing a proteomics experiment, how do you usually weigh some of these more novel techniques like ion mobility, mass spectrometry, nanopots versus the more conventional bottom-up proteomic workflow? Do you usually try to employ all of these methods if you can, or are you looking for specific applications to some of these tools? So that's a really good question. So, you know, as I mentioned, it's always the biological question that drives what technology you're going to use. So when you go with something like ion mobility, you know, maybe you need enhanced separation, you want to do it faster. So maybe then you're going to do liquid chromatography, ion mobility, and try to do the analysis that way. If you want more in-depth coverage, maybe you're going to apply more of a normal approach. If you're going to go for smaller samples, you can't get your sample masses to the necessary quantitation that you need, the amount you need, maybe you'll switch to more of our sample limited processing steps. Okay. I guess, Leanne, were you filtering any of the questions from the other?
No, to be quite honest, I didn't know the answer to most of them. They're too detailed about mass spectrometry. So there's quite a few in Zoom as well as in Discord.
I can see there are quite a few.
And if I knew the answer, I would have answered, but mostly I didn't.
I think probably keep your questions coming, and then we'll also make sure that we address them throughout the day. We like really detailed questions. And then for the other presentations, I think we'll keep an ongoing list so we can address any in real time after our presentations too. Oh, we have a new one down here. Let's see. Are those techniques related to types of samples? For example, plants and microbes? Yeah, that's a great question. So it does depend on your sample type. So if you looked at Ion mobility with those particular plant samples, we saw polyphenols were actually complicating some of our liquid chromatography, tandem mass spectrometry measurements. So by using ion mobility, we were able to move those contaminant molecules into a different drift space, so we could really confidently identify those peptides. Okay, more questions. How much time do I have, Leanne?
You
Until
have
the next
about 5 more minutes before you
5 more
jump.
minutes. Okay. I'll talk forever if you let me, so you gotta tell me when to stop. Here's a good question from Elizabeth. Is it possible to detect multiple types of post-translational modifications in one preparation instrument run using bottom-up proteomics? So traditionally, if you think of phosphopeptides, we'll cover this in more detail later today, but they're in very low abundance. And if you just do a typical bottom-up proteomic analysis, it'll be difficult to detect those. So we often perform enrichment steps. So we'll use a iron type column to make sure we pull out all the phosphopeptides and then analyze them so they're all at high enough abundances so we can detect them confidently. It depends on your post-translational modification too. With top-down, you have the ability to look at an entire protein. You're not going to get high depth of covers. You're not going to cover the whole proteome, but you can look at particular proteins and see all those post-translational modifications at once. Okay, I think since we only have a few minutes left, I guess Yu-Chin, do you want to try sharing your screen, and then I'll keep putting answers for all of these on both of our things. And also, all of us that are presenting today and later on in this week, you have our emails, you have, our contact information. If there's anything comes up, please contact us and we can address specific questions about, your research studies too.
So maybe one last question in the Zoom chat. Did you see the question about MPlex and which omics types we can study with the MPlex extraction?
Yeah, so with MPlex, so with that, it's a modified Foltz extraction. So it's chloroform and methanol. And what you get with that is you get on the top are your polar metabolites. And we usually take that off and we do gas chromatography or, LC metabolomic experiments. On the bottom, you get nonpolar lipids. A lot of those are large structural lipids that you think of making up, the bilipid membranes. So, phosphatidylcholine with its 2 fatty acyl chains and all, phosphatidylethanol, phosphatidylinositols, and some lyso species. So we'll see a lot of those in there. And then in that center layer, you get the protein disc. It works very well for a lot of different organisms. Sometimes with, our fungus systems, our yeast systems, things that have those harder kind of cell structures, we need to add extra beading or homogenization steps in front of it. But other than that, it works pretty well for many different types of analytes and many different types of samples.